Okay, this is just like a small practical session with regard to the bacterial staining. So you should have just 1 minute. Sorry, just 1 minute, people. All right, just a small problem, and then it's solved. Now can you see the screen now? Hello, can you see the screen?
Yes, doctor.
Okay, that's fine. So you should actually be able to identify all the types of bacterial stainings and describe in details the gram staining methods and also the ZN staining methods. So as you know by now that bacterial staining is one of the main ways to identify and categorize bacteria. So it's a process by which you enable the bacteria to be visualized. We have many types of staining historically. So there is like a simple staining in which usually the bacteriologists use one dye. But these days it's not very useful in bacteriology because it enables very poor identification. What's commonly used is what we call it differential staining, in which here it's composed of more than one dye, and the main aim is to differentiate between bacteria, and usually the method is multitask or multi-step actually. The most important is the gram staining and ZN staining methods. However, there is an advanced way of staining known as specialized staining, in which also sometimes you may use more than one dye. However, the main aim here is to visualize a category or a substructure within bacterial cells. Sometimes you can use this specialized staining to stain the capsule, to stain the spores, to stain certain molecules in the bacterial cytoplasm. The most important method is known as the gram staining method, which is used mainly to classify bacteria into two big groups. As you remember from the introduction slide, the two categories that we usually refer to is known as the gram positive and gram negative bacteria. This staining method exploits or make use of a difference between bacterial species in their cell wall structure, cell wall chemical composition, and cell wall thickness. That is described by this man known as Hans Christian Graham at the beginning of the 20th century. So, as I described, it is a differential type of staining method which composed of multi-step, more than one dye is used. So this is the order of all the steps and the types of stains and the materials and chemicals used in the Gram staining methods. So start with the primary stain known as the crystal violet, then iodine, alcohol, acetone, and safranin known as the background stain. What you need to understand is what is the purpose behind using each of these methods, and then how the gram positive looks like, how the gram negative looks like under the light microscope. So the principle as follows. So firstly, you apply the primary stain known as a crystal violet. As the name implies, it stain all the cell blue, as you can see from the diagram. And then you add iodine to form what we call the crystal violet iodine complex. Here, this complex will intermingle within the peptidoglycans within the bacterial cell wall, and this complex will be firmly fixed to the cell wall structure. The principle here, because the gram-positive bacteria, as you remember from the first lecture, the cell wall is composed of multiple layers of peptidoglycans. That is to say, the crystal violet iodine complex will be strongly fixed. And the gram-negative, because it's composed only of a one layer of peptidoglycans, the complex will be loosely fixed. How can we test this firmness of the fixation of the crystal violet iodine complex? We try to decolorize it. We try to remove the crystal violet iodine complex using alcohol and acetone. In the first case, in the gram negative, because their cell wall is quite thin, so they will lose their outer lipopolysaccharide membrane, the inner peptidoglycan layer, and then, as you can see, the whole crystal violet iodine will be removed because it is quite thin. So, in the gram positive, because they will be firmly fixed and trapped within the multi-layer peptidoglycans, that's why they will be or they will not be actually decolorized. So they are still look violet color. So as you can see, after decolorization with alcohol acetone, still the gram positives, they are like violet, the gram negative, they are colorless. So now we add a counter stain known as safranin, which is red. So here the non-stained gram negative become red, but still the gram positive, they are blue. And at the end, the gram positive will look violet or blue. The gram negative, they are red or sometimes pink. So this is a principle of the gram staining procedure. Crystal violet primary stain fixed by crystal violet iodine complex in the gram positive. They will be firmly fixed, firmly trapped. The gram negative will be loosely fixed or loosely trapped. Okay? How can we test this stiffness of the binding or the fixation? By adding alcohol acetone. Gram positive still will be not be decolorized. The gram negative will be decolorized. A counter stain known as safranin O, so it's red. That's why the non-stained gram negative they are red, and the gram positive still they are blue or violet. This is just the practical visualization. As you can see, you apply the crystal violet, the fiber stain for like 1 minute, and then you dispose it, wash it with water, and then adding iodine for 1 minute. You dispose it, wash it with water, and then you add alcohol acetone for a few seconds, and then you add safranine for 1 minute. You dump it, wash it with water, leave it dry, and see it under the light microscope. So these are the real picture under the light microscope, for example. So first of all, if you want to comment on a gram stained smear, firstly you comment on a color. Okay, so what is the color, for example? So just here we have two colors, either violet or blue, or sometimes because they are deeply stained, or red. So as you can see here, the color they are blue, but blue and violet is just in your mind. So here you translate it into what we call gram positive, and then you comment on the shape. So it's the coccus or it's the bacillus. Is it rounded or is it elongated? So they are gram positive coccus or cocci, and then you can comment on any extra features. So for example, here they are arranged in chains, for example, as you can see. So they are gram positive cocci and they are chain forming. Here also, firstly comment on the color. So they are gram positive because they are blue or violet. They are cocci because they are rounded. And then you can comment on any extra feature. So they are arranged in, for example, in clusters. So they are gram positive cocci cluster forming. Here also comment on the color first. So they are gram positive. Shape, they are cocci. Arrangement, for example, extra feature, they are arranged in pairs. Each two, they are clumped together. So you can see now the color has changed. So they are gram negative. Shape, they are cocci. Extra feature, they arrange in pairs. They are gram negative cocci pair forming. So here, comment on the color, gram positive. Shape, bacilli here, shape has been changed. And for example, they are rectangular, arranged in chains. So as you can see here, they are gram positive. Bacilli, they have very nice arrangement. Sometimes they arrange like a pile of matches or Chinese letters. We'll have a lecture about all of these organisms in the future. So gram positive bacilli, and as you can see, they are arranging like a drumstick. So here, gram negative bacilli, each two arranged in parallel. Okay. So gram negative bacilli arranged in chain. As you can see here, gram negative bacilli, but they are curved. Okay, looks like an English comma. Here, this is a little bit tricky. So if you look carefully, it can be mistaken as a cocci, but they are a little bit shorter. You can see gram negative bacilli, but they are short, sometimes known as a cocco bacilli. So as you can see, gram staining is a universal method for categorization of bacteria. However, sometimes certain bacteria cannot be stained with the grams method, hence they cannot be categorized by this staining procedure. Like for example, mycobacteria. This group of bacteria have large amount of lipids in their cell wall. That's why the dyes of the grams methods cannot penetrate their cell wall. That's why we have to use an alternative staining methods like, for example, ZN stain. Another group known as the spirochetes like Treponema pallidum. So they are extremely thin. So that's why they are very difficult to be stained. So the grams dyes cannot get fixed within their thin cell wall. That's why we have to use another advanced methods like dark field microscope or something we call it fluorescent staining. Another group of bacteria known as the mycoplasma. So as you can imagine, they don't have even a cell wall, and they are quite small. That's why you have to use alternative staining methods like silver stain methods. Another group known as the intracellular organisms like chlamydia and rickettsia. So they can only be used because they are extremely small and intracellular. That's why we have to stain or wait for inclusion bodies to arise to be stained by Giemsa stain. One of the famous methods for staining of mycobacteria known as the Zeal-Nielsen stain or the ZN stain. Okay, due to the complex cell wall structure of the mycobacteria. So they are waxy, impermeable to the dye because of the presence of large amount of fatty acid like mycolic acid, fatty acids, waxes, and some complex lipids. That's why they have to melt down these materials before enabling any type of staining. So in the ZN staining, so there are primary stain known as the carbonphosgene, so it's a red stain, and then we have to use a steamed heat, okay, which is gradually melt down the waxes and the long chain fatty acids. So the primary stains will enter the melted down cell wall, and then when they are cooled down, it will get trapped there. Even they will resist decolorization by strong decolorizers, and in this stage they are acid and alcohol. We try to stain background stain, which is usually blue, known as the methylene blue, and as you can see in the slide, the mycobacteria appear red against blue backgrounds. This is what we universally know as acid-fast bacilli or acid alcohol-fast bacilli. Red bacilli against blue background. Finally, there is some idea about specialized stains, like for example, in mycobacteria, we can use fluorescent stain, as you can see here, which is just antibodies linked with something we call it fluorescent dye, like uramine, rhodamine. And as you can see here, staining specialized structure within the bacterial cell wall, appearing like fluorescent green against black background. One of the famous specialized stain like spore stains. So, like for example in Bacillus, so we can use malachite green stain, and as you can see, the bacilli appear red and the spores or the endospores appear green. Also for capsulated organisms, you can use what we call the India ink stain. So everything will stain black or deeply blue, and the capsule will not get any color. As you can see, the bacilli will appear with the white halo surrounding the organism. So, some textbooks that enable you for fetching more, and thank you very much. Any questions so far? Do you have any questions? Do you have any question? Yes. Do you have any question?
No.
Okay. Okay, as you, by now, this week, we have like multiple session with regards to the introduction to bacterial general features of bacteria, like bacterial structure, bacterial classification, bacterial metabolism, bacterial normal flora, bacterial genetics. That's why I urge you to revise all of that because next week we'll start to discuss how bacteria can be diagnosed, how bacteria can cause disease, how bacteria can manifest within the clinical features, and how bacteria can be treated and prevented. Okay, that's why you urge you too much to do that. If you have any question or any inquiry, you can contact me. Actually, I prefer Telegram, and also we have like a group within the WhatsApp for further inquiry and further trade. From next week, we'll have like regular tasks and regular formative assessments, but I urge you to contact all of your colleagues just to improve the attendance on the lectures. Thank you very much. شكرا يا أستاذ شهدي، شكرا جزيلا لك.